Hello everyone, this is Aloysius Guy with you, and today's topic is very interesting one. It is about what you must know about cancer. So we will start with the tumor classifications, okay? So there are so many different classifications of tumors. Some are based on tissue types. There's also histological classifications. There's molecular classifications. Also, there are some stage classifications depending on the stage of the tumor. So here we are showing the tumor classifications based on tissue, and there are at least 5 different types. So we have carcinomas, we have sarcomas, we have leukemia, we have also lymphomas and myelomas. So now we're going to see what each cancer um, or which tissue are affected by each cancer type. So first of all, we have carcinomas. So carcinomas are cancers of the skin. It might affect the nervous system, also the gastrointestinal system, or the respiratory system. When it comes to sarcomas, they are actually affecting the muscles and the bones as well. Leukemia are known as the cancers of blood and mainly white blood cells. So there are some cancers that arises from neutrophils or eosinophils, or sometimes even basophils. So these are all known as leukemia. And then we have lymphomas. So these are the cancers that affect lymph nodes. They're also affecting glands like adrenal glands, the thymus, and the thyroid. And then finally, we come to the myelomas, which are the cancers that affect the bone marrow, and they are mainly arises from plasma cells. So so that's mainly the classification based on tissue types. So there are at least 5 different types. So to understand tumors, we need first to understand how the cell function in a healthy tissue. So what we have here is a healthy human being. So what happens is that within the tissues of the human, the cell, we would have here one cell with the DNA material within the nucleus. So what happens is that the cell will start dividing, and then at the end of this process, we will have 2 cells from that single cell, and also the DNA material will be duplicated. The whole process is called the cell cycle. So eventually, then this cell division will give us a very well-structured, healthy tissue. So tumors are mainly arise after the DNA of a healthy cell receives some kind of damage. So here, what we have here is a chromosome, okay? So the chromosome within the cell would have a DNA, and that DNA is made up of something called nucleobases. So the boundaries of the DNA are sugar phosphate backbone, and we have then the nucleobases in there. So the sugar backbone with the nucleobase are both called nucleotides. So within the body, we have 4 different nucleotides. We have adenine, we have guanine, thymine, and cytosine. So what happens is that the DNA might acquire some genetic mutations because of different mutagenesis. So, and there are different factors that are considered mutagenesis. Sometimes, some of them, for example, smoking cigarettes, also alcohol. Both of them are strong factors for causing cancers. So when we say a genetic mutation or a DNA damage, what do we exactly mean? So that's what we exactly mean. So within the healthy gene, we see that we have a G and C, and then there's a complementary C and G there. So what happens in a mutation is that there will be a change in the nucleotide, or sometimes even a deletion of a whole nucleotide. Okay, so this would result in what we called a mutation, and that in many cases might be the first step to get cancer. So the question is, what does exactly happen within the tumor tissue? So within a tumor tissue, first, before becoming a tumor tissue, we would have a healthy tissue, as we show in the previous slide. And after that, some kind of DNA damage or mutation will happen to the DNA. and this mutation will not be repaired by the repairing mechanisms within the cell, because all the time our cells are gaining mutations all the time, every day. However, the body and the cell can repair these mutations before they become cancerous. However, in some few cases, the cell cannot repair the damage anymore, and this will result in forming a tumor cell. So this will convert the cell to become tumor cell. So when we say a tumor cell, we are talking about a cell that show very high and excessive rate of division and growth. And as we can see here from this image, we can see that the healthy cells will be outnumbered by the cancer cells. And that's mainly what's happening within a tumor. So this amount of cells will form eventually a lump, which is we call benign tumor. And within that lump, we would have healthy cells with tumor cells, but as we said, that the tumor cells will be far higher in number, and over time the body will start forming a fibrous capsule that encapsulates the whole tumor cells within that area, and this will make the tumor very restricted, and also in that case the tumor is easier to treat. However, as we can see here, that the tissue outside that capsule, they are considered healthy tissues, and they are usually referred to as tumor adjacent tissues, and they contain healthy tissues. So as long as the tumor is confined within there, everything it's less harmful and it can be treated. So the second type of tumors, which is the most dangerous one, and it's much far more dangerous than the benign tumors, they are called malignant tumors. So what do we mean by saying malignant tumors? So as we saw before, that when we have a cancerous cell, it might stay like that for so long, forming the benign tumor. However, there's a chance that over time of the growing of the benign tumor, these cells will acquire even more mutations that makes them even more aggressive and makes them even more dangerous. So this results that throughout the time and throughout accumulation of all these mutations, the cancer cells, there's a possibility that they will be able to break through the fibrous tissue. So what happens then is that once the tumor cells break out through the fibrous tissue, which means that now they are entering the healthy tissue region, which is, we talked about, that the region surrounding the fibrous. So this means that if we are talking here, for example, about a liver or any organ, at the beginning we would have a very small patch of that or small part of that organ that is cancerous. However, after this happens, it means that an even bigger part of the organ will be cancerous now. And all cancerous tissue are non-functional tissue. So the bigger this region is, the more non-functional the organ is as well. So that's why it's always best to identify cancer when it's still benign. That's why all doctors recommend to do regular checks, because if there's any problem, the doctor might identify cancers in that stage. However, if it is in this stage, the treatment will be much harder. The second thing that might happen is that these cancer cells over time acquiring more mutations and making them more aggressive. So these cells, the cancer cells, will induce the blood vessels to enter within the tumor. And once the blood vessels enter there, the cancer cells will be able to go through the walls of the blood vessels and then move through the bloodstream and go to distant organs. For example, if this is the pancreas, so they might go through the blood and go to the liver then. So by that, you would have a cancer or the patient would have a cancer in 2 different organs. So in both cases, it's very dangerous, and that's why it's called malignant tumor, and they are the most dangerous ones. So now we need to understand which genes are most commonly mutated in cancer. So there are 2 main types of genes that are usually associated with cancers, and the first one is called proto-oncogenes. So what is proto-oncogenes? So first of all, we will study the normal function of proto-oncogenes, and then we would go to the mutated function. So within the DNA, there's areas called genes. So a gene is an area of the DNA that produces a protein, okay? So some of these genes are called proto-oncogenes. So the question is, what type of proteins are produced by proto-oncogenes? And the answer is that those proteins produced by proto-oncogenes are proteins that stimulate the growth and division of a cell. So as we can see here, if a cell starts producing that protein, it will be induced to divide and increase in number. So the question is, what happens when a mutation occurs, which is cancer associated? So first of all, a mutagenesis like that occurs to the gene, including factors like alcohol and cigarettes. So these mutagens might induce a DNA damage within the gene, and this will make the gene become an oncogene. So the name proto-oncogene refers to the healthy gene, and oncogene refers to the mutated cancerous gene. Okay, so once the oncogene become cancerous, there are two main possibilities that might occur. The first thing is that this mutation or DNA damage will result in producing a higher rate or increases the rate of protein production. So as we can see here in normal conditions, this protein stimulates growth, but it's produced at a very regular and regulated levels. However, in cancer, it will be just upregulated, and there will be a lot high expression of that protein. The second thing that might happen is that this mutation will result in slightly changing the activity of the protein and makes it even hyperactive. In both cases, this will result in making the cancer cells dividing much faster than the normal cells. And here are some very hypothetical days and time periods. Like, for example, if the normal cell might take 3 days to produce 2 cells, the cancer cell might produce, 6 cells or something, or 4 cells just in 1 day. So that's the problem of oncogenes, is that they upregulate the expression of proteins that induces cell proliferation and growth. So the second type of genes that affect cancer are called tumor suppressor genes. They are commonly associated with cancer. So as before, we will go through the normal and mutated functions. So as we said, that the DNA has many areas contain genes, and some of these genes are called tumor suppressor genes. So the proteins expressed by tumor suppressor genes are usually proteins that inhibits or stops cell division, and they induces cell apoptosis. And cell apoptosis, it means just induces the cell to die or to self-destruct. So what happens is that once the cell starts expressing these proteins, they will inhibit cell division, and also, or they might induce the cell to self-destruct. So, for example, sometimes a DNA mutation might occur in the nucleus of in the DNA within the cell, and that DNA mutation, it might be unrepairable. So what the cell would do is that it will induce a self-destruct to prevent that cell from becoming cancerous. So they are very important in regulating the function of healthy cells. So what happens when they are mutated? So a mutagenesis might occur in the gene, and if it is a tumor suppressor gene, this will prevent the production of those proteins. And this indicates that when the cell do not have that protein anymore, it means the cell will just keep dividing, and this will increase the number of cells, and also this will inhibit cells to self-destruct as well in some cases. And that's also why tumor suppressor genes are very important. So when a mutation occurs in the oncogenes, this will induce the cell to divide, and when also mutations occurs in tumor suppressor gene, it will also prevent them from not dividing. That's why if we keep cancer over a long time period without treatment, these type of dangerous mutations might accumulate, and this would make the cancer even far more aggressive. So now we need also to understand and highlight that these mutations that occurs in oncogene and in tumor suppressor genes, they can be acquired, which means they occurred to you or to the patients through environmental factors, for example, like alcohol and cigarettes, so they induces DNA damage. Some of them are hereditary, which means that they were passed down to you from your parents. So now we need to understand how each of these mutations occur in the body. So first of all, we would have the germline mutations, which means that these mutations occurred in the sperm or the egg of the parents, okay? And we have somatic mutations, and this means that these mutations occurred in the embryo. And that's a very big difference between occurring the mutations in the sperm or egg or in the embryo. And we will see why. So if the mutation occurs in the germ lines, like sperm or the egg, then the embryo will carry the mutation, and then the entire organism carries the mutation. And this would lead eventually that 50% of the children, there's a 50% chance of a chance that the children or the offspring of that patient will carry the same mutation as well. However, if it is a somatic mutation, then only a specific part of your body will be affected by that mutation. And since the mutation is not in the germ lines, it will not be passed down to your offspring. And that's how the genetic mutations are passed down through generations. And also that's why it's important, for example, in breast cancers that are commonly passed down from the mothers to their daughters. It's very good to do regular checks as well to see if you have mutations in specific genes like the BRCA genes, and see the chances of passing down the mutation to the offspring. So now we come to understand the strategies and the methods that are used in cancer therapy. And these methods and strategies are depending on what type of cancer you have and whether it's local or systematic cancer. So when we say local cancer, it means that the cancer cells are localized at a specific region of the body. And if the cancer is like that, then surgery and radiation will be one method to offer to the patient for treatment. And usually for benign tumors, that where the tumor is very confined in one and restricted in one area, surgery is enough to cure the patient. However, for systematic patients, you are usually used when the cancer just go throughout the body and leaves the primary site. And in that cases, they are usually, as we know now, they are called like malignant tumors. And in that case, there are chemotherapies, some like using drugs, hormonal therapy, and also targeted therapy, like immune therapy. And each one of these therapies, I might do future videos on them, because each therapy have a lot of different drugs in them and a lot of different targets, but this is like the general strategies used for cancer therapy. And here are 3 questions that I believe they might test your understanding of the tumor process and the tumor progression. As these information that are included in this tutorial, they are very essential and they are must-know information about cancer. And now we get to the end of this tutorial. I really hope that this video was easy to understand and follow. And please don't forget to like, comment, and subscribe, and also to hit and activate the notification button so you can get all of our new and future videos. Thank you for watching.